Oh, that's nice. All right, everybody. Hang on. I'm just tidying up my workplace, so to speak. Do okay. So, all right, I'll get everything kind of sorted while people roll in. Hope you've all had nice lunch. Felt like I had to scarf it down a little bit faster than I probably would have wanted to, but that is what it is. So, this is lab, right? It's going to be a little bit weird because let's get back. Yeah, that's fine. Um, Obviously, we're not doing this in person, and that poses, some issues, obviously, because a lot of what being a scientist is about practical skills, stuff you do with your hands. But really, to be honest, that stuff is something that anyone can learn easily in whatever context, half a brain kind of thing. So if you're good enough to move on through the major, getting a C or better in kind of every one of the courses that you need to, then you're more than smart enough to be able to pick up those kind of manual practical skills. The thing that is really important to learn, and which is actually far, far harder, is the stuff up here, right? How do you analyze the data that you collect? How do you design the experiment so that you collect the right data, or how, asking the right question, right? Once you've got that data, what do you do with it after that, right? So these are all really important skills, and really that's what you're going to be getting from this lab. Well, not this one today, obviously, this course, right? This lab course. And that's really what I've put a lot of kind of thought and energy, I guess, into, is getting you all of those skills and experience, which are really, in my mind at least, the important things. And then, once life goes back into normal and teaching goes back to real life stuff, then you can build on that in other labs, right? But you're still going to get the full analytical kind of theoretical, like mental experience that you would if you were attending lab in person. And really, the reason why we're doing this online is because the restrictions on how many people you can have in lab at any one time, and the fact that we're doing this kind of crazy, like twice the speed sort of schedule, would make it next to impossible to actually do the lab as it's designed, right? So you'd end up with a worse experience in my mind. I mean, last semester during spring, it was kind of crazy most of the time, and a bunch of stuff didn't work because we were trying to fit it into this weird and wacky schedule where people attend alternate weeks, and I just couldn't see a way of doing that over the summer and actually have stuff work reliably, which is kind of important if you're going to get out of it what you're intending to. So that's really why we're doing this stuff online. It's, I think, gets you most of the benefit, and also, you know, without the inconvenience of having to come into campus to actually do the lab. So even though it's going to be a little bit weird, I did this last summer, uh, and it worked pretty well. I think people got as much as it was possible for them to get out of it, and pretty much what I intended. So all of these labs are going to be held in this format online. Same kind of deal as with the lectures. So as me, chuntering away. Zoom sessions will be posted to YouTube. Anything that we, cover in terms of like drawings or chat or whatever will also be posted to Blackboard. Pardon me. So you have, access to that. Now, obviously the big difference is you're going to be doing this stuff online, right? So, hello. Just talk about some really important stuff, Mr. Robert. Nothing to worry about. And to get around that, essentially, I'm gonna have you do really, normal lab, right? Probably easiest to explain this way. Normal lab, you go to course content, you go, lab 1 today, right? You download the lab handout, it would look something like this. And it would have a kind of an intro, an overview, talk about some of the stuff, details about what you're going to be doing, and how. And then you'll have stuff to do, right? So normally, well, actually, I didn't do it this way last semester because it worked out pretty well online. But, standard format would be you would fill these things out on a piece of paper and then you'd hand it to me the following lab, right? And so instead, what we're going to do is all of these questions are now online for every lab it looks like a quiz because that's like what it looks like, but really it's just the questions that you have in your lab handout. Chat is blocking that. So for each question where you need to calculate something, I give you the information that you need to calculate, right? So these are the things that normally you would collect yourself or things that you would do yourself. So I'm going to give them to you. Sorry, just going to bounce around a little bit and yeah, I'll come back to this in a second. So for example, lab 3, which will be next Tuesday, right, you would be normally running a electrophoresis gel to separate DNA. Now, obviously we can't do that because I don't want to give you these things to do at home just in case bad stuff happens, I guess. And so instead, I'll give you that gel picture, right? So you miss out on the actual loading of a gel, watching it slowly go down the gel and then taking a picture of it, which is kind of a shame, but you'll get other opportunities to do that. But you will still get the gel picture. This is a rather nice one that a student took at some point, me teaching this. And you'll still be able to go through all of the activities, answer all the questions that you would normally if this were in lab, right? So for example, and here's, stuff I do to explain something, right? And so I'd take a picture of that. I'll delete this one because we'll be making ones of our own. But that stuff goes up onto Blackboard in the folder for the lab in which we covered that. And so, for example, in this one, and they vary a little bit, right, from one place to another, right? So here I give you the gel picture, so that allows you to do the work. But the difference here is that you're going to draw a graph and then take a picture of it or save it as a JPEG or Excel file or whatever, right? I don't really mind. You could draw in crayon if you like, as long as I can see what you're doing and you follow these requirements, right? So in every question, I'll put down what I expect you to do. It's drawing a graph, label the axes, right? Kind of what is it that you're supposed to be doing? And then you upload that file and I'll grade it by looking at whatever you uploaded and giving you the points for that particular exercise, right? So that's one kind of, pardon me, way of doing this, I guess, one kind of format, I guess, where you're going to be uploading something. You also need to give me the values or the data that you calculate using those graphs. And again, every lab, I'll go over this with you, right? So this isn't like remember this shit because I'm not going to talk about it again, kind of deal, right? I'll go over this next week when we talk about this lab. But here you enter things that you calculate using those graphs, right? Whereas in the lab today, for example, which is a good example because it's today, instead I'm going to give you the data in the questions, right? Which is essentially the vector, which is a series of data points. And then you're going to work through the lab and calculate what you're being asked, right? Okay, so what's the mean? I want it in centimeters because that's what I'm giving it to you because that's what scientists work in. Want the value to 2 decimal places, right? So it might be 166.12 centimeters, right? If you don't give it to me to 2 decimal places, I might get a little cranky and like knock a point off. But I'm not intending to be, a jerk about any of this stuff. The aim is for you to get as many points as you can, right? You just have to do it, right? And so do it as soon as you can. I'd actually recommend doing this the day of the lab, so this stuff's all fresh in your mind. Also gives you time to figure out if you didn't understand something, look at the YouTube video maybe, send me an email if you don't understand, because you have until midnight the day of the next lab to submit this. So for today's lab, you have until midnight on Thursday to submit this, right? And so this is all about just doing, calculating things, yada, 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 right? Oop, doop, doo. And so these are like auto grade things, which is like fabulous from my point of view. But they're pretty straightforward. Then at the end, I want you to draw these data as a graph, right? Because showing or putting data into a format which is readily accessible is a very important skill as a scientist, right? So you have to be able to turn this into something that you can actually use to explain whatever you found and what it means, right? You can't just give someone put in a paper, oh, this was the height vector, ta-da, it's different or not, as the case may be. You have to put this into format. So there's going to be a bunch of graph making in this course, both for these individual weekly labs, but also for one of the bigger projects, which is going to be the fly linkage project. Anyway, so this is the general format, right? So every single, go away. Every single lab, so 1 and 2 this week, 3 and 4 next week, so on and so forth, right? Has its own assignment typically, right? So for example, Mendelian genetics, right? You got to do some chi-square and stuff. All the information is provided for you in the handout, right? Other ones such as the linkage project don't. Better get rid of that because I'm actually going to be kind of asking you to do an analysis of some like a bigger data set and then write a lab report based on that. So it kind of varies from week to week. Generally speaking though, if you want to know what's due when, as always, don't ask me, look at the schedule, right? So this again is posted here. Syllabus is always useful, right? It will tell you what various things are worth in terms of points. Schedule's the most important one, right? Because, you're not going to change the points, right? They are what they are. This is basically laying out everything that we're going to be doing and when different things are due, right? So this tells you, okay, Thursday, I need to go to lab 2 microscopes. I need to look at whatever he's going to be talking about then. And that's where I'm going to find the assignment that I'm going to do. If there is an assignment for that one, there may be, I can't remember. No, there isn't because there's nothing in the assignment box next to it. That's an easy way to find out. And it'll also tell you when the different things are due, right? So generally speaking, you have stuff to do for most labs, right? Not every lab, obviously, but most of them you have stuff that you need to submit. Those deadlines in the, these, all right, these things, essentially your digital or virtual lab handout submission. These all have their own deadlines. They're all available now. So technically speaking, you could do like the entire semester's worth of lab in 1 week if you want to kind of bash it out. I wouldn't recommend it, but you can if you want to. These will all have deadlines and will close according to whatever it says in the schedule. So the pipetting one is going to be due midnight Thursday 17th. Another good part about that, and actually changed things a little bit after last summer, is that if there's something you don't understand about what we're doing today, for example, you have the chance to ask questions in person if you don't want to do it by email or whatever, next lab on Thursday. All right, so before we finish and kind of get on with the rest of our afternoon, you can actually say, "Hang on, I didn't actually really understand what was the graph that we were supposed to do. Like, what are you interested in? Like, what's the things that you're going to be looking for to assign a grade, right?" And so there's actually the opportunity to do that in the next lab session before that assignment is due. And again, that's another good reason for trying to do this stuff the same day as you actually take the lab. I know you're going to be tired, and there's probably lots of other things you want to do other than genetics. And, I'm kind of in the same camp, but there's stuff that you have to do. And I would suggest doing this the same day, right? Because if it doesn't make sense, then you have, a couple of days with which to at least to sort it out, including in person in the next Zoom session for lab. So that's the general idea. Each one of these, yeah, I think that's about right. Each one of these lab assignments is worth 50 points. The Eugene, which is a bioinformatics software, which is really cool, but kind of hard to use. That assignment's worth 100 points, right? So every lab, really every week, you'll be able to, again, as with the homeworks in the lecture part of the course, you'll be just able to add to this stuff, right? And so don't leave any of those points on the table. They're there for the taking, right? And it's designed that way so that you can keep, building up towards your grade, even though that grade then gets transferred to your overall course grade, like every single week that you're doing this, right? And generally speaking, roughly like back the napkin kind of calculation, students typically do 2 grades better in lab than they do in lecture. Not always, right? There's always exceptions, but people always do better in lab than they do in lecture, just because the exams are hard, right? And it's a tough material to fully get your head around, right? So make the most of the lab. It is very much intentionally designed to help you, not just in becoming, better scientists and trained biologists, but in getting a good grade in your genetics course, right? So don't fail to answer any questions or not do any lab assignments, because you'll just shoot yourself in the foot, right? The other 2 things which are big assignments, I guess, would be for this linkage project. So this would actually be a genetic cross that you would do if this were in person with actual flies, and you'd collect your own data and you'd generate your own results or graph and lab report from that. Obviously, you really don't want to be doing this at home. Like this isn't like electrophoresis. This is a house full of irritating fruit flies, right? Because lab is always full of irritating fruit flies after we start this. So I'm going to give you those data, right? And we have a bunch of different support ways in which you can get help to analyze that. Obviously, we're going to be going over that in person. There are videos that my predecessor who taught this course made, which are very good. And the output, like what I want from you, is one, a pretty graph in Excel. So where you turn those data into a visual representation, essentially. Right, and that's a very, very important thing to do. And then taking those results and then writing a mini lab report. And I'm talking like 2 to 3 pages, like double space kind of mini lab report. But with the formal structure of intro, methods, results, conclusion, stroke discussion, right? And with a bunch of things that you need to analyze in that results section. And so if you go to, it's all here. It's like a treasure trove, genetic stuff. Right, go here, this file here really tells you everything that you need to do. Tells you what is worth what in the, we'll go over that later, that's fine. In terms of a rubric, like what I'm looking for in the lab report, which different sections are worth what. And again, we'll go over this at least once, if not probably twice, before that's due. But those two things together, the Excel file with the graph and then the mini lab report, they're together are worth 300 points out of a total of 800. So again, do a good job of that. I am very happy to give feedback so that you can improve your lab reports, which makes my life better because then I get to grade better lab reports. As long as you give me enough time to do that before the deadline. Now, because it's all like kind of squished together, that makes life a little harder because there's less time to do it. So also look ahead as well, be proactive. Anyway, so this basically tells you what we're doing and what is due and when. And you'll find everything that you need to know to do the lab in each of these folders, right? They're all very deliberately designed to give you kind of what you need. We're also going to have a few like PowerPoints, like kind of lectures for the lab part. Those are also posted in those lab sections, so you can get a jump on that if you want. Go through those or wait till we go through stuff in the lab. It's going to be kind of weird, but also I think in many ways, it's kind of a plus too, right? Because certainly from last semester where, I had a lot of people who were absent from lab or didn't necessarily understand it first time round. A lot of people kind of struggle, particularly with the math heavy stuff, right? And so because we're doing this all online, all of this is going to be recorded, so you can just go back over and work over stuff, right? If it didn't make sense first time round, as if you were studying for an exam. So in some ways, it's actually better. Not in all ways, but in some ways. And so that's pretty much it. There's no lab final, right? So there's no exam for lab. All of this stuff will be, when's the Eugene stuff due? Yeah, so that the Eugene assignment is due what would have been the lab after the Eugene lab. So midnight of Thursday, 29th of July, right? Which is kind of after classes finish, but before grades are due. And that's pretty much it, to be honest. One thing that I'm actually always pretty excited about, whether online or in person, this linkage project is neat. It's actually kind of more neat to do in person, to be honest, which is a little sad. But this project, essentially what we're going to do is we're going to, as if we were working kind of in a lab, right? So this is figuring out the inheritance of two genes. We're then going to take that as if, you know, we'd done a forward genetic screen, like a mutagenesis screen, let's say. And we're going to analyze the gene itself, right? And so this is a kind of step-by-step, almost a simulation. Actually, if you're doing it in person, it wouldn't be a simulation of what you would do in a research lab if you wanted to characterize a particular mutation or a mutant allele, right? And so you'll be going through the whole process, right through from kind of like what the actual phenotype, all the way through to the molecular information in a bioinformatics software program. And it's actually kind of cool. I was talking about, and this is a series of experiments that's recently been accepted for publication in the American Biology Teacher Journal. So it's like a teaching journal. So it's actually going to be published as a paper, kind of what we've developed over the last, well, I guess, 4 years now as a project. And it's really neat. And I was talking about this with the Dean of the College of Arts and Sciences, kind of about this paper. Oh no, actually, I was probably complaining about how tired I was after trying to teach Eugene in 2.5 hours rather than 5, which is what a normal semester would have. And she was really impressed. She's a physical chemist, I think is probably the best way of describing her field. And I said, oh yeah, and we use this open source bioinformatics software called Eugene to do some bioinformatics and genetics. And she's like, wow, that's pretty cool. You know, where I came from, Texas State or Texas Tech or wherever it was, we never did anything that advanced in genetics. So it's actually pretty cool what you're going to be doing. And I'm really excited for this one. And again, it's not quite the same, not doing it in person, but it's still, you'll still be doing all the same things as if you were in person. So that's pretty much it for like the general format of the lab. Again, your lab grade as a percentage gets converted into, or basically I take 25% of that and then add that to 75% of your lecture grade. That gives you your course grade. So it's fairly straightforward to work out. Whatever, you know, if you get 100% in lab, then you get 25% towards your overall genetics course grade. If you get 50% in lab, you get 12.5% towards your overall genetics course grade. So it's not necessarily going to make the difference between you getting a C and an A for an overall grade, but it certainly does make the difference between you getting a D and a C or a C and a B. That has happened many, many times. Okay, so this is really something that you want to stay on top of. Actually, all of this whole genetics course, lecture and lab, it's all about staying on top of your shit, because if you don't, you'll get buried. Right? It is and especially so in summer, because this is just, it's actually, I'd be fairly comfortable calling it insane. Right? It's a really, really intensive experience learning genetics in 8 weeks. It's enough of an experience doing it in 16, right? Students rarely come out the other side going, oh, so glad I took genetics. Well, I think they are glad. Maybe they'll appreciate it a little bit more once they've had a little bit of time to decompress and relax, right? And you're going to be doing that in 8 weeks in half the time. right? So keep on top of all of this. And if you're ever hit a wall, not literally, obviously, but if you kind of get stuck on something and you're like, oh shit, I really just don't want to do this. I don't understand it. It's just like drives me crazy. Don't let it slide. Contact me. Dr. Crook, I just don't understand how to do this thing in Eugene. You showed it to me and I just can't do it myself. Fine, no problem. I'll help you. We'll go through it one-on-one if necessary, right? Because if you kind of hit that stumbling block and you don't get over it, then the next couple of days later, you'll have another one due, right? And then another one, and then you'll have an exam, and then you've got homeworks, and it's just non-stop, right? So if, obviously, I hope you learn a bunch of genetics from this course because it's a cool subject and I like teaching it. But what you're definitely going to get, particularly if you're successful in this course, is how to manage intense, heavy workload courses, right? How to stay on top of your stuff. And that's a skill that will last you throughout your whole life, without a doubt, but certainly will help you when you start doing other kind of more challenging, or maybe equally but differently challenging upper division courses for the rest of your degree, right? So think of it from that perspective too. You're getting a very intensive lesson in time and, schedule management, I guess. So really stay on top of that. And if you feel you're stumbling for any reason, don't let stuff slide and think that you'll get back to it to another time, because another deadline will come up. You'll have a homework that you've got to do. You're going to have an exam that you've got to study for. You're going to have another lab that you have to submit, right? You will be buried. And I really don't want that to happen because it's very hard to get back from that, right? Because there's just no let up until we stop on the 28th, 25th of July. There's no let up whatsoever. It is full on. Okay, so stay on top of your lab stuff too. That's a really, really successful way of getting the grade that you're hoping for in genetics, or at least the grade you need. Maybe you're not going to get what you hope for, but you'll get what you need, which is what's important. Okay, so does anybody have any questions about the general structure of the lab, what we're going to be doing, how we're going to be doing it, anything like that? I think the coffee that I drank earlier is just kicking in, which is good because I'm starting to hit my 2 to 4 o'clock, I want to have a nap slump. So I have to power through that. Okay, so just, yeah, that would be nice. So basically what we're doing here is based on the fundamental concept that as a scientist, you can never sample everything. So whatever you're doing in terms of collecting data, whatever that might be, you'll never be able to sample the whole population, or almost never. And even if you do, that population is going to be a subpopulation of a larger population. So that's one issue, right? That you're going to have to deal with. Like, it doesn't matter how many data points you record, you're not going to be able to record all possible data points out there, right? You're not going to be able to sample the whole population. So we have to deal with that statistically, right, in kind of terms of quantifying stuff. Second issue is biology is noisy, right? It isn't, and this is why chemists and physicists and particularly mathematicians tend to kind of look down their noses at biologists, right? Kind of grubbing around in the mud kind of thing, because biology is noisy, right? It is a highly complex, highly variable, multifactorial process where lots of different things can change what you see. And because of that, you know, whether it's in like an internal difference, like within an individual, or an external one in the environment or whatever, right? You will get variability or variance. And so whenever you collect data, you have to figure out how much variety, right? Basically, pardon me, there is within your samples and between your samples, right? Because essentially what you're trying to do is find out, is the difference between my populations bigger than the difference within the populations, right? And so really what this lab is about, in a fairly straightforward way, is trying to get a handle on that stuff. And we'll come back to that later when we look at like probability and statistics, when we look at some of the stuff for the fly linkage project, when we're going to be using our chi-squared test. And you'll certainly come into this a lot in other biology courses, right? This is stuff that I do pretty much in terms of research on a day-to-day basis, right? Whenever I'm doing any kind of analysis, right? So this is very much kind of ground level basics, but those are basics that you'll keep and use over and over and over again throughout the course of your career, hopefully. I'm just going to rub off my lovely worm picture. I really want to figure out where my other markers have gone. Kind of annoying because my wife has a habit of tidying up my office, but then that just means I can't find stuff. So it's all going to be in blue today, which is a bit of a pain. I don't know where my red and green pens have gone. But here you go. Look at that beautiful whiteboard. So essentially what we need to do is, let's say we have, we're calculating collecting distribution data, right? So we have height here in centimeters. Just make sure you can all see that. And we have number of people, let's say, right? So this is the number of people that have a certain height. Now, automatically or automagically, as you would guess, height is a non-discrete measure, right? So you can't either be tall or short. You're going to be a height on a continuous scale, right? So you're going to have something that looks like this, right? So you're going to have a collection of data points, right? And if I had a different color pen, oh, they were tidied up, but they were tidied up in a way, didn't bend my neck around far enough. All right, I'll take back what I said about my wife. Thankfully, she's not within hearing range, so. Ah, isn't that beautiful? Mr. Roberts probably, shit, I can't actually see what he's doing. Hopefully that's not too small. And so these will be your raw data. Let's say we've got two different populations, might have two different sexes, maybe two different treatment groups. One were given one drug, one given placebo, whatever, doesn't really matter, right? And so if I were to show you these data, it would be very hard for you to figure out, are these different, right? It's just a collection of data points. So really, oh, I do have my camera there. We need to go from that to either something like this, or depending on the type of data, right? It really varies. And there's also a lot of art, I would say, about how to best visually represent your data. So if any of you are particularly artistically inclined, there's potentially an awesome career ahead of you as a scientific illustrator. And those people are very, very much in demand because most scientists absolutely suck at art, right? Me included. Good at other things, but you know, pretty pictures is not one of them. Or it might be something like this, let's say, right? Really just depends on what kind of data you're working with and what kind of story you're trying to tell, right? But you need to go from your raw data to a summary version is probably the best way of describing it, right? And so you have to do some kind of basic analysis of these data to convert them into this representation without hopefully losing too much of the information stored in these data, right? And that's kind of the tricky balance you have to strike, right? Because anytime you summarize something, obviously you're reduced. Damn it, I need to buy a new camera. There we go. You're going to be reducing the complexity of what you show, right? So there's a risk that you'll actually lose stuff. And so a lot of what we're going to be talking about today is how do we do a good job of summarizing those data in a kind of like a descriptive way without losing too much information about that data, so we can draw other conclusions about what we're actually seeing, because it's all well and good like making pretty pictures. I'm doing this because I'm actually rubbing the board off as I speak. How about that? Mad professor skills. Shit, what was I saying? I forgot. Anyway. You just... Oh, that one's easy. That's extending the extra warranty on my car. Can even recognize that from the area code. So essentially we'll want to take a data set. We want to do some descriptive statistics, like mean particularly, or median if we have some non-normal data. We want to figure out how much those groups vary from each other, or really vary within, and then we look at how much they vary from each other. And then we want to display that in a way that is visually accessible, right? And that being the key, right? Because that's how we kind of get other people to interact with the data that we've collected, right? And that is not a trivial process at all. And that's kind of getting back to that whole, if you're good at art, then being a scientific illustrator or a scientific kind of interpreter, I guess, is actually a pretty cool career to have. I actually only ever met one in my however many years of doing what I do. But it was really cool. So that's essentially what we're going to be doing today. And so really whenever you have a kind of, actually, I'm going to just stop sharing so that this becomes a bit bigger. That might make it a little easier to see what I'm doing of. And again, kind of this pokey little whiteboard isn't ideal, but it's better than me gesturing wildly, right, with my hands. And so
going back to your analogy real quick, sorry to interrupt you, about data being noisy, right? Are you essentially trying to get something that's more raw and you're trying to make it less noisy by creating into one of those like bell curves graphs that you were describing, or or showing us?
Both, because you still need to know how noisy those data are. And I'll kind of draw, actually, that's a really good point. I'll draw you a couple of different graphs, right? You see, I wasn't kidding when it comes to talking about artistic talent, right? I could draw some cats really well. So these two look the same, right? I don't know. Let's just call this, right? I don't know what measure this might be. Propensity to shout at other drivers when you get cut up, maybe. Whatever, it doesn't really matter. Now, if I put on, so these are the means, right? And so with means, you don't have any information about the noisiness of each group because it's just a single number. It's the mean. Now, if I were to put standard deviation bars on here of so, are these two groups different? Are these two groups different? So this gets back to noise, right? So really, if we want to see if two groups are different, whatever they might be, this is just a silly example, right? What we're asking really is the variance between the two groups larger? Oh, thank you, Rose. Is the variance between the two groups larger than the variance within the groups? That's kind of like the formal way of saying it. The other way of saying it is, do the error bars overlap? Because these error bars are really a mathematical measure of noise, depending on exactly what you're doing and how you want to portray that noise. So if the noise within each group, say in graph A, if that noise is smaller than the difference between the groups and the means are different, right? Essentially, you have a region. Yeah, I apologize for my handwriting. It's pretty terrible. There's a region where this error bar and this error bar don't overlap, right? So it doesn't matter if there's noisiness here and there's noisiness here. That noisiness is not greater than the difference between them. And so we can be pretty confident just by looking at this. Obviously, there are statistical tests you can do to formally ask that question. But we can pretty confidently look at that and go, yep, these two are actually different, whatever these two things are, right? Or whatever the manipulation that you're doing is causing that difference. Now, in these ones, the variance within each group is greater than the difference between the two groups. Or in other words, the error bars overlap. I've kind of exaggerated it for, purpose sake. You can technically have that and it still be significant. It just depends on other stuff, right? It kind of depends on the design experiment, the statistical test, your statistical power. But again, as a very rough and ready, like quick look, you could easily look at this and go, yeah, if they are different, then that difference isn't very big. Or is another way of looking at it. There may be a difference, but my statistical power, the sample size, let's say, is too small. So if this was a sample of, I don't know, 20 drivers from rural Oklahoma, right, where maybe you don't see another car for several days, it's not going to be enough. If you looked at 2 million drivers from San Antonio, which seemed to be a crazy bunch, generally speaking, then this variance may shrink and you may get to the point where you can make some kind of inference from the data. So you have to have that measure of noise. What's your first name, Mr. Robert? Is it actually Robert?
Yeah, no, yeah, it's Robert.
That's okay.
My first name. Yeah.
So. I didn't know if it was you had a first name as a last name kind of thing, which is totally fine.
Well, I had a professor call me Mr. Robert at one point, so I actually changed the name to Mr. Robert. I guess it just transferred over from my other class. I was like, oh, dang. So
that's fine.
I guess I'm Mr. Robert again.
So that's why it's such a good question, Robert, because whenever you take that cloud of data points, okay, Zachary, fine. There are more than one car in Oklahoma. I've never been there. I don't actually know. Whenever you take that cloud of data, those individual data points, the raw data, right, and you process it so it's more manageable, you need to maintain a measure of the noise, because that's really important. And actually, if you look at any kind of statistical test, that kind of varies from test to test. But when you're looking for a difference between groups, they almost always require some measure of variance or noise. And that varies, obviously, by sample size. So there's usually lots of different things that go into a picture like that, right? But you have to maintain some kind of measure of how much that whatever it is you're measuring varies within each group as well as between the groups. And that's really what we're going to be talking about today, right? So the very first thing that you want to do is take those and turn it into a mean. And that's, this is actually a lot harder than it looks, promise. There you go. Right, that's the symbol used for a mean. It's an X with a line over it, right? And really what you're doing, and you all know what a mean is really, is that you're taking all of these points and your mean equals, x1 plus x2 plus x3, however many of these are, you add those values together and you divide them by the sample size. That's it. Nothing more than that. Right, and that gets you your mean. And damn it, I've got a very cantankerous camera. Here we go. And that's how you take that kind of cloud of data down to a number of points which you can plot. Right? However, when you get that single number, you lose all of the information about the noise in this sample set, because you just end up with one number, right? So you might have thousands of individual data points and you convert that to one number, you lose all of that information. Does that make sense? Right? I mean, you need to turn into a mean because there's no way that, yeah, you can't really plot the raw data. So you need to have, first step is calculate the mean. Or, and that's with a caveat, if your data are normal, right? And so normal equals bell curve, basically, right? So you expect a normal distribution. If we measured everybody in my neighborhood in terms of height, you would get a few people who are really short, a few people who are really tall, and most people will be kind of in the middle. That's essentially what normal distribution is. Or median. Now, if you measured the height of, I don't know, everybody at a Lakers basketball game, you'd get most people are normal, and that would be the basketball team over there. They're really, 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 really tall, right? And so you wouldn't get a normal population. So you use instead the median, right? Median is very useful for dealing with weird outliers, right? Because those weird outliers, particularly if you're working with a continuous variable like height, let's say, really skew the mean hugely. You know, so if you have a bunch of people who are, I don't know, 150 centimeters tall, and then you have someone who's 250 centimeters tall, that's going to move the mean way further to the right than it should be. And the median equals the middle number. Right, so if you have 1, 2, 3, 4, 5, median is 3. If you have 1, 1, 2, 3, 4, 5, the median is 2.5. It's not the mean, right? So that's kind of a way of dealing with non-normal data. There are other ways of doing it, which I am not the person to ask about because statistics is not my strong suit. But this will do for now. So that's the first step, right? That gets you your single point, which you can plot, right? You don't have to plot a distribution. You can simply plot a number. All right, anybody got any questions? Questions. Other than my lack of experience of driving in Oklahoma. I've never actually been there. Kind of want to, just because, you know, it's not that far away, I guess. Although everything seems kind of far away in Texas, to be honest. Right, so you calculated the mean. Step 2. Is calculate your variance. And variance is a very specific thing. So there's also, pardon me, that's my lunch paying me a visit again. These things. No, there's no I in mean. These are how you will portray the noisiness that goes with that mean. Right, and so these are very formal terms, and we'll go through what they mean in a little bit. But this, so whenever you want to, step 3 will be plotting this, right? You need both the mean and the measure of noise, whatever that might be. Now, we don't actually ever use variance for reasons I'll get to in a second. We either use standard deviation or standard error of the mean. Sometimes you've probably heard of confidence intervals, right? Which is essentially your confidence that the number falls within a certain range around that mean. That's another way of expressing that. It's not something I use a whole bunch, to be honest. Kind of depends on the data that you're collecting. And so, best way of describing variance is, let's say you have your mean here, right? And we have 3 data points, let's say. That'll do, makes life a little easier. Your variance equals the sum of the differences between each data point and the mean. So this data point down here is going to contribute a lot to the variance because it's very far away from the mean. This data point here is going to contribute relatively little to the variance. But the mean has to be represented by all of the variance. And that's why you have to add it together for each of those. Now, you can look at the second one. I don't know if that's clear enough. Might need to draw bigger dots. You can inherently tell that these data or this data set is less noisy than this one, right? You know, it's kind of visually obvious, right? But we want to turn that into an actual number that we can plot on a graph, right? And so the way you do that, and I'm going to be taking a lot of pictures today. This is so much easier when you've got a whiteboard that's bigger than 3 foot by 2 foot. So this is variance. You'll probably already see an issue with it, but we'll get to that in a second. This is the sum. That's what that funky E is. I think that's epsilon, maybe. I'm not really good at Greek letters. Which is, I don't think it actually matters which way around it is, to be honest. The mean minus, say, one of your data points, right? Again, we're talking about how far each individual point is from the mean. Don't worry, we're nearly there. Tell you a sneaky trick. You can do all this stuff in Excel. Super, super easy. I've got to give you the theory so you actually understand why you're doing it, but I don't do any of this stuff manually. I do it in Excel, right? And it's sum because you're going to add these together. So we can kind of forget that E for now. Oh, give me just a second. Now, that's not squared, that's x2. Maybe I should put it down here. That's probably better, isn't it? There you go. Now it's like the sequel to X-Men rather than X-Men squared. Now, the problem with this is that because it's the mean, you're always going to have points that are above the mean and that are below the mean, right? Because otherwise it wouldn't be an average. Now, whichever way round you do this doesn't actually matter because you're looking for a difference. But one of these will result in a negative number. So if you add a positive number and a negative number, you end up with 0 or not the sum. And so you have to square all of these, right? And then because it matters how many times you do this, let me just double check. I'm pretty sure this is n minus 1. This kind of shows the fact that I don't actually do this. Oh yeah, it is n minus 1. There we go. Make sure you can all see that. And there's a statistical reason for doing that. I just can't remember what it is right now. I kind of learn it every semester and then I forget it as soon as I taught it. It's basically to represent this fact that you're not sampling the whole population. That's kind of the short of it. So in this case, you're looking at two different points. It'll be 2 minus 1. So this would be divided by 1. That's how you work out variance. And you can do it by hand. You can do it kind of sort of in Excel. You can also do it in Excel with the variance function. Problem is, if you're calculating the variance of height, you're squaring the units of height. So you'll end up with centimeters squared, which is area. And that kind of sucks. You don't want that, right? So what you have to do is take away that squaredness, right? I'm just going to have to take a picture and rub this off. Bless you. That's either someone sneezing or someone dropping something, one of the two.
Yeah, sorry, I unmuted myself on accident.
No worries. It's funny, my wife's doing training for an online job, working for the COVID hotline, and she actually has heard twice people that haven't muted their mic taking an interview for another job. How embarrassing is that? Okay, so we don't want to use variance because variance is squared, right? So we want to use standard deviation. I'll force myself to write it out in full. Shorten to SD, and that's not South Dakota. That stands for South. Call myself out. That stands for standard deviation. So there is no like ND, right? And what that is simply put the square root of the variance. Or if you want to write it out in full, the square root of the sum of that squared n minus 1, which is variance. So all of that we've already covered before on the previous whiteboard. You just square root it. Questions? This is quite a lot to take in. It's like kind of a lecture in statistics after having 2 lectures of genetics. Thank you, Dipper. Honestly, people I work with can't get the staff nowadays. Okay, so that's one measure of noise, standard deviation. Now, if you are doing this over and over again, instead of, let's say, you're repeating in South Dakota, you're repeating in North Dakota, there you go. Then you now have 2 different samples. Maybe you do it again in Oklahoma, you'll have 3. So now you have a repeat measure, and you want to take that into account when you look at the noise, because presumably, pardon me, the more populations you sample from, the more confident you are that this is like a universal thing, right? So this isn't something that's specific to one particular subsample or subpopulation or one particular area or species or whatever. This is applicable to all of them, right? And so now you can be more confident about the mean that you generate. And really that's what this is about. It's about like the confidence in the data that you're collecting. Or maybe instead of doing it in different places, you record, I don't know, songbird density over successive years, right? So whatever you record in one year might be due to other things that you're not measuring, say, average rainfall, or if it's a La Nina or El Nino year or whatever, right? And so if you do it in successive years, you can be much more confident that the result you get is reflective of what you're actually doing rather than something you're not taking into account. And so to take that into account, did I take a picture of this? I can't remember. Nope. Good job I checked. You know, Dipper, I'm just going to have to remove that from the whole chat box before I post it up, you know, so it doesn't exist, kind of thing. Hope that's all right. So the SEM, I'm not going to write that in full because it's just up there. All that is essentially giving you a bonus for how many times you repeat this measure, right? And so that's standard deviation divided by the square root of the sample size. So if you're, you know, you repeat this 4 times, right? Your SEM will be half of the standard deviation. If you repeat it 10 times, it's going to be, oh shoot, what's that? Let's say 100 times, that's easy to work out. If you repeat it 100 times, it's going to be 1/10 of the standard deviation, right? And so essentially that's kind of really, the bigger the N equals, the greater the confidence. Are you being kicked out of there, Robert? Like so. Yeah, I think he was. That's a bummer. All right. And so really what you want to do after all of that, right, and this is a very, very long way of kind of getting to the point, is you want to draw a graph. There you go. And so that is back and is on the move. You're in cab, aren't you? I recognize that style of window.
No, actually, I'm in the new building behind the cab.
Ooh, yeah, very nice. I've never actually been in there.
I was trying to take over a room, and then I got kicked out. So I was like, oh, okay.
Yeah, that sucks. Well, I guess you know not to use that room in future, right?
Yep, noted.
Okay, so now we've gone from this kind of massive data, right? Now we have our means, right? And you can draw those as lines. You can draw them as bars. You know, it's up to you. Kind of depends on, again, what story you're trying to tell. And now you can draw, if this is plus minus 1 SEM, now you have your mean. Right, which is the dot or the line or whatever that you're drawing then. That's going to be plus 1 SEM. So that's essentially your mean plus the SEM value, or the SD, if appropriate. Right, and it really varies depending on your experimental design. We can always get into that another day. And this is going to be minus 1 SEM SD. And so, and you do the same for the other one too. And so this is how you portray the noise in your samples. And noise is really, as you can see, because every one of these functions involves the sample size. And so really what these are saying, these are measures of confidence. How confident are you that your mean lies within a particular range? If you repeat this, your mean should, like with the same conditions, the same population, same everything, your mean should exist between these, the second one, between these two bars. Right, so this is really a measure of confidence on how accurate is your mean, how accurately does it represent what's really going on in that population. Does that make sense? And so as you can imagine, the bigger the sample size, or the more times you repeat this experiment for the same population, or maybe different populations and doing the same thing in different places, the more confident you will be of your result. So as your sample size gets bigger, essentially your confidence gets greater, and so your margin of error really gets smaller.
How come we can do, oh, I'm sorry to interrupt.
No, go. You should never be sorry about interrupting, Marissa. Just ask a question whenever you want.
Cool. So I was just wondering why the EM and the SD are like, why it can be either or, and how would we know which one to use? And
that's a really, really, really good question. So let's say, what's the best way of okay, so kind of thinking of a good example analogy. Shoot, you know what? I'm just going to use what I do for my research. So I work with C. elegans. It's a tiny little nematode. So it's an animal, but it's an invertebrate. It's a really good model organism, not organ. Pardon me. Now, if I were to get a mutant, right, in a particular signaling pathway, let's say, and I was to score a particular phenotype, now I'm going to have a population of animals, and I'm going to pick animals from that population, and I'm going to measure them for whatever, lipids in my case, right? However, all of those individuals are largely the same. I mean, they are genetically identical. The only variation you're going to get is based on, like, if you pick the same age, or if they are just fed, or stuff like that, right? Even if you were to look at other plates, because they grow on agar plates with bacteria, you're still looking at the same genetic background. Does that make sense?
That does make sense. So
okay, so just give me a second. So I'm kind of like building up to the exciting part. So if you were to collect a bunch of data from that population, right, that one genetic background population, you would calculate the noise by standard deviation, right? Even if you were to repeat that experiment, you're still sampling the same kind of, not the same individuals really, but the same population. So in that case, you would use standard deviation. Now, if I were to, let's say, do that on different food, right? Same genetic background, different food. I'm not actually interested in the food. I just want to kind of rule it out as a potential variable, right? So now we actually have different biological replicates. Now, if I did that a bunch of different times, now I'd actually have essentially a biological replicate And so now I could use standard error of the mean. So if you're sampling from the same population without changing any variables, you would use the standard deviation because the population is the same. You're above a certain point, you're not going to be able to be more confident of your result, no matter how many more you measure. Because really your mean and the standard deviation is your measure if you didn't sample the whole population, right? Even if you sampled half of it, it's still going to be a sub-sample of the population. Does that make sense?
I understand up to the part where you're saying like with the food and the animals, so two different types, then you would want to bring in the standard error of mean, right?
Yeah, so the standard error of the mean is really if you're doing a genuine biological repeat. And so if you're growing a kind of particular strain of wheat, let's say, right, and you are interested in a type of like effective fertilizer, right, on growth, whatever bushel size, something like that. So if you had a very large field of wheat and you sampled from different points kind of in that field, right, which are all treated the same, you have what are called experimental repeats, right? So you're just kind of repeating the same thing, but from different areas, right? So that you would use standard deviation for. Now, if you were to repeat that the following year, and then the year after that, now these are genuine biological repeats. Now you're taking the same set of conditions, right? And you're repeating it again in like a very much a distinct, discrete population. So no longer you sampling from different areas of the same population. You're technically speaking, you're sampling from different populations, one from year 1, second from year 2, third from year 3. And so now you essentially have a more robust experiment. Does that make sense?
Yes, that does make sense now with the different sets of data. Okay,
right.
Thank you.
And so, yeah, and so really then if you have a more robust experiment, so whatever data you get isn't just because of something you're not taking into consideration. Let's say, I don't know, average sunlight levels, let's say. Now, because your experiment is more robust, it kind of controls for those other variables, right? Even if you don't know what they are. Now you can have more confidence. And so now you can reduce the standard deviation kind of by, or your measure of noise by the square root of your sample size. Right, so if you do that 3 times, now you can reduce that kind of measure of noisiness by 1.74, so square root of 3. That's actually often my sample size, which is why I know. Right, if you do 100 times, right, not necessarily over 100 years, but maybe 100 different sites around the country, now you reduce your standard deviation even further by a factor of 10. Right, so if you get whatever result in all of those different places, now you can be super, super confident that your actual population mean, right? Because you never measure the whole population, you just measure a part of it. Your actual population mean exists between those 2 bars. So essentially, this is what we mean, right? So this is the mean of your sample, of your subpopulation. And this is your kind of confidence that your actual population mean, like if you were to measure every single stalk of wheat, right, within an entire field, your mean should be somewhere within these 2 bars. And so the closer those bars are together, the more robust measure of the population you can say you have, essentially. Does that make sense?
Yes, that does make sense now, and when to use those. Thank you.
Yeah, so I mean, it's a good question. It's a very good question to ask, actually, because you want to be very careful with any kind of statistics, whether descriptive or analytical or whatever, not to do something inappropriate, right? Where you use a measure which isn't justified because it makes it look better. Right? You have to be very, very careful of that. It's very easy to fall into that trap. And so asking those kinds of questions is a very good idea. What's the appropriate measure to use? And so I know what it is from working with worms because I've been doing that for years. But whatever you kind of come across individually, then you need to ask that question pretty much every time. And so what I'm going to do very quickly is just show you some tricks, right? So I use Excel all the time. Actually, I was just doing some analysis for another student of mine, this stuff here, right? And so these are actual data from RNAi experiments, so RNA interference experiments, knocking down these genes in worms and looking at survival of particular cells which die by necrosis. Always make my ears ache. And so pretty much I'm doing exactly the same thing as what I'm teaching you. These are 3 distinct biological replicates. So you calculate the mean of each of these, calculate the sample size. I've calculated the standard deviation. And really, when I get around to it, right, which is probably tomorrow, because these are 3 biological replicates, they happened on different plates over a number of days and so on. The appropriate measure would be to use a standard error of the mean, in which case I would divide this by the square root of 3, because I have 3 biological replicates. And so what I've done here, and I'm not sure this is the exact same data set, I probably just made them up quickly, is to show you that you can do this in Excel, right? You can actually do it as in the handout by hand. And obviously when you have a small number of data points, that's manageable. If you have thousands, it is not, right? And so learning how to use Excel is actually a pretty cool thing to do. And I'll show you some neat tricks. And so wherever you have Excel, you want to have your data for different groups, females and males in this case, this is supposed to be height, in the same format, right? So we've got both in the column, right? You don't have one in the column and another one in the row because that would just mess things up. And then you want to use functions, right? And so again, I'm probably teaching some of you to suck eggs here, and that's fine. You can just kind of switch off. But if you haven't done this, this is like super cool, right? So if you want to access the functions, hit the equals button. Now you got here. What you'll see is this is the cell number. As soon as you put equals in, it brings up a function. And this is a little drawdown menu, right? So I want to work out the mean, also called the average. Now you select that, right? So I'm just going to go back again because it doesn't hurt to show these things a few times because I'm used to doing this. It doesn't take me any effort, but I want to do things slowly. So equals and then select average from this drop down menu. There are actually more functions, kind of lots of things you can do in Excel. But now you have, it kind of auto suggests a range, right? Which in this case, and you can click here, is this range here, like so. Bugger it. It's already selected that. So you can change that. You can actually write in here. You can click OK, right? And if you, there we go. If you actually click on the argument at the top, the whatever, it will show you what data it's using. So if you want to calculate the average for all of them, if you very carefully go over the square at the corner, click on it and drag, you can select other data too. So this would be the average of all data points, right? So once you're happy with whatever you've selected, just hit return and it calculates it for you, which is really cool. Median, again, equals median. Now you see the issue here is that it can't distinguish between your actual data and the fact that you've calculated the average, right? And so this argument is wrong. You can see now what it's going to be doing is calculating the median from your, what would that be, 7 data points and your mean, which is incorrect. So let's hit OK, even though we know that's wrong. And now we can just drag that up. If I can find, I've got to tell you with a touchpad, it can be a real pain in the butt. I'm just going to draw that, drag that up there. There we go. Kind of a sloppy ass way of doing it, but it'll work. Okay, so that's how you generate your median. And your median should be, if you have a normal population, it should be pretty close to each other. Variance, I actually need to, I think I can remember how to do this. I never calculate variance because we never use it, right? But it's worth doing. And now you can go more functions. So if you go back here, equals, more functions, and let's type in the box variance. Think. And think this is variance of the sample. So for example, here you distinguish between the variance of population, which we are not sampling because we're sampling a subset, and variance of the sample. Okay, so I don't want all of those, I want these. I think that's right. Or is it P? Ah, fudge it. We can work out what's right or not. So standard deviation is also easy. Again, another function argument. Again, just drag and drop. Hit OK. Gives you that. And the standard error of the mean is the standard deviation divided by the square root of the sample size. Now you can do this in lots of ways. If you want to be like super fancy about it, I can't remember. Let's see if I can remember how to do this. This is going to be the standard deviation divided by the square root. Yeah. I might not be able to do this properly. Let's see. As they say, even a blind squirrel finds a nut every so often. Nah, it's not interested in that. Okay, so it's good. Oops. I thought I remembered how to do that, but I don't. It's kind of a little, you can calculate n separately, which might be a good idea, but we'll just do it this way. Oh, I didn't do divide by, did I? Dumbass. Maybe I could have done this right the other way. So if you want to put things inside, like actually work out, let's say the square root of 7. No, that's not working. All right, fine. I thought I could remember how to do this, but maybe I didn't. I actually had it from the previous class, but I deleted all that because I thought I could remember how to do it. So let's just do it the easy way. 7 square root is 2.646. There you go. Feel like a bit of a Luddite. 646. Fine, as they say. Now, if I had time and mental function, which I don't right now, I could probably figure that out. Maybe I'll do that afterwards. So that's working it out for essentially one group, right? And what you've got over here, this is working out the variance for each individual point, right? So this is the sample point 1 minus the mean squared, so on, added together gives you your variance, which is 230.95, and that's the same as the variance you get from the sample variance, which is a good double check. Now, the cool thing, if you keep everything in the same format, copy those. So again, that just, those little wandering ants tells you you've copied something. Copy, or you can go to copy. I use keyboard shortcuts, so that's like Control C, and paste that. Yeah, just paste it. What it does is it pastes the formulas, and so as long as your other data points are in the same format, it will automatically copy that for you. How cool is that? So that means you can save a ton of time. You don't have to enter these things all over again. You can simply copy the formulas over and do it. So again, you can do this manually with a calculator and a piece of pen and paper. You can also do it this way too. So even though you're spending longer in lab, like listen to me chunter on, you should hopefully spend less time afterwards filling out the lab handout. And so the last thing that you want to do is, and this is copy. Now, if you want to paste those data elsewhere, now you're going to have to do a special paste, right, which is the down arrow, and you want to just paste the values. Otherwise, it will try and calculate the mean from those cells above it, and that's not going to work. And we want, what did I ask you to do in the, is it SEM or SD? Probably SD, I would guess. Let's see. Make sure I'm telling you the right thing. No, SEM. Control C. Forgot what I was telling you in the beginning, didn't I? All right, so now you want to make a graph. Insert graph. That one looks nice. You want to make sure you have axes labels on this, axes titles. So this would be, yeah. Sex, height for that one. Get rid of that. And now what you want to do is you want to do your error bars. And again, we'll be doing this for the fly linkage stuff, and you'll probably be doing this for other things too. But this is a good kind of dry run. So if you click on one of the columns, right, it shows you what you've plotted, right, over here in the colored boxes, but it also shows you that you've highlighted that. Now you go back to chart elements. You can also right click, right? So I think it's that plus button there, depending on what version of Excel you have. Select error bars and add more error bar options, because there's obviously lots of options. That allows you to kind of fancify it, right? But what you want down here is custom. Click on specify value. And now it'll ask, what's your positive value? You select those two, hit return. Your negative value is the same because it's plus and minus. Do the same and click OK. Now, obviously, you might want to change that a little bit, maybe not. Now you have your mean, right, which is 161 for females and 166 or thereabouts for males. and you have a measure of variance of those two populations. And so now you can kind of look at that and go, yeah, the height of the males and females is pretty similar. That is how you go from raw data to something where you can go, oh yeah, that's what's going on, right? And so those steps are the way in which you do that. Now, so depending on the type of data, and Marissa, what I'll do is I'll actually go back and double check what I told you, because that applies to the work that I do. I'm not 100% sure it applies to other types of analyses, right? And I don't want to lead you astray because I'm not a statistician. I kind of know really well the stats that I do for the work I do. I don't know very well the stats other people use. So I'll check on that, add it to my list. And if I was wrong, which is possible, I'll let you know next time we talk in the lab, because I don't want you to get the wrong information, because that might cause you issues later.
Okay, thank you.
Yeah, no worries. I mean, the fun thing about teaching, one reason why I really enjoy teaching is because I learn a lot of stuff as a result. You know, it's quite easy when you do research to kind of become like pigeonholed and bunkerized in one particular set of knowledge, I guess. And when you teach, you can't do that. You kind of get forced not to. So that's just an example of that. And that's pretty much it. It kind of went on a little longer than I promised. Sorry about that. I just want you to get a really good kind of understanding and feel for what we're going to be doing, because when you do this for the fly linkage work, this will be a lot easier and quicker for you to do then. Also, you can do it all manually. So if you want to draw this out on a napkin, as long as I can read it and see that you've done all the things you do, take a picture of that, post it up, and you'll get the same points, right? So I'm not really, I'd prefer if you'd use the cool software, but I don't mind if you don't either. And that preference is really just because it's easier. Sometimes I have actually had people do stuff on napkins, and although that's fine, it's not the clearest medium with which to work sometimes, particularly if they use it to wipe up ketchup or whatever. Has anybody got any questions? You're probably thinking you want to be somewhere else right now. If you do have any questions that occur to you as you do it, either email me or save them for lab on Thursday, and then we'll spend a little bit of time at the beginning of the lab going through it. Again, don't worry if this stuff in Excel didn't make a whole bunch of sense. You don't have to use it. You can do the lab handout for today, the lab exercise, quite perfectly fine without it. But if you did want to use it, it's a good skill to learn, and it's a good way to save time, even though you kind of have to invest time in learning in the first place. It's kind of a trade-off. All right. You're all probably brain blasted, and my ears are certainly aching from my headset. I need to go borrow my wife's. It looks way more comfortable. It's like a big gamer headset or something. And that's pretty much it. So all of this stuff will go up online. Probably going to have a coffee first, to be honest. I'm pretty pooped. This afternoon, evening, so I don't forget. I'll send you an announcement when it does, and I'll include in that a YouTube link to the playlist so that as you go through this later, you can just refer back to that as and when necessary. That's pretty much it. Whew. First day over. We've only got another 15 to go. It'll fly past, I promise. All right, everybody. It's been a lot of fun. I shall see you on Thursday. Take care.